A warm welcome to all. Trinity Cultural Academy Coimbatore, in collaboration with NEPAS New India Publishing Agency New Delhi, is launching a new YouTube channel called NEPAS. Is providing videos for agriculture and related programs. And these videos are highly useful for ICAR All India Entrance Examination, Junior Research Fellow and Senior Research Fellow for PG admissions, and ICAR NET National Eligibility Test, ARS Agriculture Research Services, and STO, that is Senior Technical Officer T6 grade. And also this video is highly useful for agriculture field officer exams being conducted by Institute of Banking Personnel Selection. And this video comes under heads and subheads are plant science, that is seed science and technology. And in this we have already given a video, that is seeds and its structure. And in this video we are going to see about sporogenesis, gametogenesis, fertilization and seed development. And this material is prepared by Dr. K.Vanangamudi, formerly Dean Agriculture, and also he was served as a Dean Adiparashakti Agriculture College and Professor and hence Seed Science and Technology in TNA Coimbatore. Let us enter into the subject. The knowledge of modes of reproduction and pollination is essential for a plant breeder because these aspects helps in deciding plant breeding procedures to be used for the improvement of crop species. Choice of breeding procedure depends on modes of reproduction and pollination of crop species. What do you mean by reproduction? It refers to the process by which living organisms gives rise to offspring of similar kind or species. In crop plants, the mode of reproduction is of two types as sexual reproduction and asexual reproduction. The first we'll see what do you mean by sexual reproduction. The multiplication of plants through embryos which have been developed by fusion of male and female gametes is known as sexual reproduction. All the seed propagating species belongs to this group. Before entering into the sporogenesis and microsporogenesis etc., we'll see the ovule, how ovule develops and what are the different parts involved here. And at the end of this lesson, you will be able to see and explain the ovule development, and you will be able to describe the Megasporogenesis and Megagametogenesis and Microsporogenesis and Microgametogenesis, and you will be able to understand the double fertilization and the seed development pertaining to embryo and endosperm development and maturation. We'll see how ovule develops. Here, the development of ovule occurs within an ovary which provides a location for nurturing and development of female gametophyte. Its sexual fusion with the male gametophyte and embryo development, survival and eventual regrowth. And the ovule growth begins as a small outgrowth within the nucellus and it continue as Megasporogenesis and Megagametogenesis continue. The region of nucellus, that is to become a ovule, enlarges and differentiated into definite morphological characteristics. Secondary outgrowth or callus, that is integuments, soon appear around the periphery of the nucellar outgrowth and envelop it. The first picture shows that the region, that is the longitudinal section of the ovary showing the developing ovule. The second picture shows that the latest stage of ovule development where the integument differentiates into outer integument and inner integument and the further development of ovule takes place. The third picture shows that the still later stage of female gametophyte showing a complete embryo sac, that is the antipodal cells, polar nucleus, synergids, etc. And the final picture shows that a generalized diagram of complete flower showing the location of a ovule. And this consists of inner and outer integuments and ultimately become the tester of the mature ovule. The next one, the different parts involved here are funicle, hilum, chalosa and raphe. The developing ovule is commonly attached to the placenta by the funicles. The scare on the ovule made where the funicles detaches at maturity is called as hilum. The next one, the point where the integuments meet at the nucellar apex is the micropyle, and the region which is opposite to the micropyle is known as chalaza. It is a region of integumentary origin. Between chalaza and the hilum of many species, an area called raphe is present. These parts are already explained in the previous video. Kindly see the first video. The next one, nucellus. The nucellus provides the tissue for the origin and the nature of female gametophyte from archisporial cell to the mature megagametophyte. It originates from ovary tissues and provides site for archisporial cell origin. Subsequently, part of it becomes trapped within the integuments as the ovule develops. Normally, no further growth occurs as the nucellus is at least partially consumed since it supplies a nutrient support to the developing embryo sac. However, in some species, it undergoes a considerable development and contributes substantially to the storage tissues as a perisperm. The next one will see the integument. The nature and thickness of the integuments vary considerably among the species depending on their role in contributing to embryo sac and ovule development. In Umbelliferae, the inner integument persists. In compositive family, most cells of both integuments are absorbed, leaving only a thin layer of crushed and disorganized integumentary tissue on the inner side of the pericarp. The next one will see the micropyle. It's an integumentary pore or opening in the ovule through which the pollen tube grows to fertilize the egg cell of the female gametophyte. The micropyle may assume one of the several configurations depending on the closure of the inner and outer integuments. The next one is epistase. It is the development of well-developed nucellar or integumentary tissue in the micropylar region of the seed of certain species in where the cells adjacent to the micropyle may show a marked elongation. The next one will see how sexual reproduction takes place. That is, in that there are two major processes, sporogenesis and gametogenesis. Here, the sporogenesis deals with the production of microspores and megaspores. In anthers, microspores are formed through Microsporogenesis, and in ovules, the megaspores are formed through Megasporogenesis. The period of let us see what is gametogenesis, and after that we'll see what is microsporogenesis in detail. Gametogenesis deals with the development of male and female gametes in the microspores and megaspores. Friends, clear about that microspores are developed from anthers and megaspores are developed from ovules. This is very important point. The next one is microsporogenesis. Already we have seen that microspores are developed from anthers. Clear about it. The next one, the period of flower development where the stigma is ready to receive the pollen is known as anthesis, and it is a time where pollen begins to blow and pollinate the female organs. The pollen is usually produced if 4 sacs or microsporangia of the anther, although occasionally fewer sporangia may occur. Within the sporangia, certain cells become, this is called a sporangium. Within the sporangia, certain cells become microspore mother cell, and this microspore mother cell undergoes two-step reduction division, that is meiosis or microsporogenesis, to yield 4 microspores, each of which microspores are haploid in nature, and each of the 4 microspores is normally functional and undergoes two division known as Microgametogenesis, and which gives rise to microgametophyte or mature pollen grain. This we'll see in later. And here, what does microsporogenesis means? In microsporangia, few of the cells become microspore mother cell or pollen mother cell. Each PMC produces 4 microspores, and each 4 microspores are functional and undergoes division and develops into a pollen grain. This is enough here. The next one we will see the microgametogenesis. Here, each microspore mother cell within a microsporangium divides to form a tetrad of microspores that soon separate. The nucleus of each microspore then divides into a tube cell and generative cell. Here, these are developed formed within the wall of the microspore, which is then converted into a pollen grain. Following pollination, the pollen grain generates and producing a pollen tube, and the generative cell gives rise to 2 male gametes. This and all we have seen. On maturation of pollen, microspore nucleus divides mitotically to produce a generative nucleus and a vegetative or tube nucleus. It is generally released during the binucleate stage. This is called pollination. Generative nucleus undergoes mitotic division to produce 2 male gametes or sperm nuclei. And the pollen along with pollen tube possessing a pair of sperm nuclei is known as Megagametogenesis. And the pollen tube enters the embryo sac through microphyte and discharges the 2 embryo nuclei. This is how a pollen grain develops. The next we'll see how Megasporogenesis occurs. Megasporogenesis. In angiosperms, we have seen mega means ovule development, or in ovary megaspores are produced, we have seen. In angiosperms, seeds originate from meristematic tissue of the ovary wall called ovule primordial, where ovule development takes place. In species with simple ovaries, these primordia are usually located near the suture of the ovary wall, where a carpel is fused. In species with polycarpellate ovaries, the seed form at the fusion of the carpels or along the septa or central carpel axis, depending on the type of placentation. In tomato, a well-developed placenta arises from which many ovule primordia develops. From the nucellus, one cell develops special characteristics that distinguish it from adjacent cells. As this cell increases in size, the nucleus becomes larger and its cytoplasm grows denser in preparation for cell division, that is archisporial cell. This cell is called as archisporial cell. The first division results in megaspore mother cell and a parietal cell. Usually the parental cell remains undivided and soon deteriorates. However, in some species it undergoes a further division and contributes to sheath formation. A diploid megaspore mother cell is formed having the same number of chromosomes as a parent plant. It undergoes two-step cell division called as meiosis, results in 4 megaspores, that is haploid cells, each having one of chromosome complement of the mother cell. Normally, only one megaspore is functional while the other 3 are degenerated. Here, the first picture shows that the location of the ovule development, that is the ovule primordia where ovule development takes place. The second picture shows that the cutway section of the lower region of the ovary wall showing origin of archisporial cell. We have seen the archisporial cell now, that is archisporial cell. Note that it is larger than the other cells, surrounding cells, having a larger nucleus and denser cytoplasm. The third picture shows that the cell division occurs during Megasporogenesis, that is 2 cell division occurs. We have seen that no 2-step cell division, that is meiosis occurs. Meiosis 1 and meiosis 2. The fourth picture shows that a cutway section of lower part of the ovary showing a location of 4 4 megaspores, 3 of which of normally degenerate and only one is functional. The next one, what do you mean by megagametogenesis? The development of female gametophyte, that is embryo sac, from the megaspore mother cell is known as megagametogenesis, which is a process of successive nuclear division within the enlarging cell that becomes an embryo sac. 3 successive mitosis occurs resulting in 8 nuclei. Here 3 successive mitosis occurs. It results in 8 haploid nucleus. Then they arrange themselves within the enlarging embryo sac and cell walls form, resulting in 3 antipodal cells at one end and 2 polar nuclei at the center and fuse to form a diploid and diploid nucleus and egg apparatus consisting of egg and synergids at the other end. That is 3 antipodal cells at one end, 2 polar nuclei at the center and the egg cell at center, and finally 2 synergids at the other end. The egg cell is a haploid one, that is nucleus with surrounding cytoplasm enclosed in a thin wall or follicle. The egg cell is positioned near the opening, that is micropyle of the ovule formed by the developed surrounding integuments. A small vacuole may be present near the point of attachment away from the micropyle. Here we have seen that 3 antipodal cells are arranged in opposite pole and 2 synergid cells on one pole and central cell and polar nuclei at the center. 2 polar nuclei and egg cell is called as secondary nuclei, and megaspore develops into a mature female gametophyte is called megagametogenesis or embryo sac. This picture shows that here 3 normal mitotic nuclear division occurs, leading to one large cell enclosing 8 nuclei. Later, the cell walls encloses the nuclei and the entire structure become a female gametophyte or embryo sac. This is called embryo sac. A mature female gametophyte consists of antipodal cells at one end and synergid cells and synergids at the other end and 2 polar nuclei at the center. The next one, fertilization, occurs. Before that, we have here the pollen grain and the embryo sac is ready. Now is going to fertilize and to form a seed. Fertilization is also known as syngamy, can occur when both male and female gametophytes are fully matured. This usually occurs in a dual fusion process known as double fertilization. When the pollen grain lands on the stigma, it germinates by sending out a pollen tube which grows down the style through the micropyle. This is called micropyle region. Through the micropyle. This is how a pollen tube develops, and here the micropyle present through which the pollen grain enters into the embryo sac and with the tube nucleus closely following the tube apex downward. The tube nucleus soon degenerates but the 2 pollen sperm cells enter the embryo sac and one sperm cell fuses with the egg nucleus that to form a diploid one, and the other sperm nuclei fuses with the polar nuclei to form an endosperm nucleus, and this is called a double fertilization. Here we have seen that when the pollen grain enters on the stigma, a pollen tube develops through which 2 sperm nuclei enters into the embryo sac through the micropyle region, and when the pollen tube grows through the micropyle, two sperm nuclei discharged into the embryo sac, and one fuses with the egg cell to form a diploid zygote, and the other one fuses with the two polar nuclei to form a endosperm nucleus. One fuses with the egg cell to form diploid nucleus, and the other one fuses with here via the microfile region. Tube nucleus enters into the embryo sac. One fuses with the egg cell to form a diploid nucleus, and the other one fuses with the polar nuclei to form. One fuses here with the egg cell, and the other fuses with the polar nuclei. There are two sperm nucleus. We have seen that now via the microfile, the tube nucleus, that is sperm nucleus, enter into the microfile, and one fuses with the egg cell, that is to form a diploid 2n number zygote, and the other fuses with the polar nuclei which is present at the center, form a 3n endosperm nucleus. At the final stage, 2n zygote and endosperm nucleus is formed. The next one, seed development. After the fusion of male and female gametes, zygote develops. After that, it will undergoes further development and form a seed. It begins with the minute embryo sac, which with certain exceptions, it is about the same in size, shape and arrangement. In spite of initial similarities, the seed develops according to the genetic specification for each species, which are coded in the nucleus chromosomes of each cell. The embryo sac may be ellipsoidal, elongated and or variously bent in shape. The longitudinal axis extends from the chalaza to microfile, and through the antipodal cell, the endosperm nucleus and the egg apparatus. Morphologically, the microfile is at the upper end of the embryo sac. As embryo sac is growing, it requires a constant nutrient supply which is provided through the chalaza, establishing a polar gradient from the antipodal to the micropilar end, as obtained from the nucleus, nucellus and integumentary layers directly through the walls of the embryo sac. And the nutrient is also obtained from the nucellus and the integumentary layers directly through the walls of the embryo sac. The next one will see how embryogeny takes place, that is how embryo forms. After sexual fusion or a syngamy, a brief period of reorganization occurs, during which the large vacuole adjacent to the zygote gradually disappears, with the zygote cytoplasm becoming more homogeneous and nucleus large. The duration of this period varies among the species, but it is usually about 4 to 6 hours before the zygote begins to divide. Lines of polarity in preparation for future division and growth already exist in the embryo sac, have been established in the unfertilized egg. The still undivided zygote typically elongates along the horizontal axis, and small vacuole becomes eventually distributed through the cytoplasm. There are 4 stages of embryo development. The first one is globular stage, and the globular stage of embryo development precedes the cotyledon development, and the embryo here is spherical in shape, and the suspensor also develops along the embryo. We have seen that the zygote develops, a reorganization takes place, that is 2-cell embryo and suspensor cells are developed. In globular stage, the embryo is spherical in shape, and suspensor, this is called suspensor cell. This also develops along the embryo. In the hot stage, the next stage of embryo development is hot stage. During this stage, the globular embryo develops into 2-lobed form and looks like a heart. Here is a heart stage. The suspensor acts as an umbilical cord providing nutrients to the embryo. The next one is torpedo stage. In this stage, the cotyledons and axis elongate, enlarge and grow longer. embryo may remain straight and become curved. And finally, the mature embryo. As the embryo matured, cotyledons curve. As the embryo matured, the cotyledons curve and provide nutrients to the plant enough to get plant started with photosynthesis. And suspensor disappears when the cotyledon enlarges, and here there is no suspensor. Here suspensor is getting disappeared, and endosperm has been used for nutrients for the development and maturation of cotyledons and embryo axis. Next one, endosperm development. The endosperm is the principal nutritive support for the embryo of many species, especially monocotyledons, during both seed development and maturation. In angiosperms, the endosperm normally originates from triple fusion of sperm cell from the pollen tube within the diploid polar nucleus, following fusion of two polar nuclei of the embryo sac. Therefore, it is a nuclear complement as triploid endosperm. In gymnosperms, the endosperm is normally haploid since it develops from one cell of the female gametophyte. It provides nutrition for the developing embryo, therefore its composition is compatible with the embryo needs. But endosperm must also draw its nutritive support from the embryo sac and surrounding tissue. This net effect is to surround the embryo with rich nutritive tissue from which it can draw for development and growth. This creates a competition for nutrients both within and outside the embryo sac. There are different types of endosperm development. It is nearly 3 types of endosperm development. Division of primary endosperm nucleus yields the micropylar and chaliceal chambers, one or both of which may contribute to the mature endosperm. When only one develops, the other is crushed and soon degenerates. Endosperm development may be of one of three types depending on the sequence of nuclear division and cell wall formation. The first one is cellular endosperm. In this type of endosperm, each nuclear division is accompanied by cell wall formation. Here there are different cell walls are formed for each nuclear cell division. Then in case of nucleus endosperm, this endosperm type is characterized by nuclear division accompanied by cell wall formation. The nuclei unaccompanied by the cell wall formation. Here the nuclear division is accompanied by the cell wall formation. In case of nuclear endosperm, nuclear division is unaccompanied by the cell wall formation. Nuclei may free or may be later be separated by cell walls that forms in one of the 3 ways. That is, 1 to 3 layers of the cell wall may form around the periphery within the free nuclei inside, or a cell may form in the micropylar area with the rest remaining in the free cell state, or the entire endosperm may be filled with the walled cells. And all 3 endosperm conditions may exist in the same family. The next one, here we can see the primary endosperm nucleus in nuclear endosperm. Next one is helobial endosperm. The helobial endosperm is intermediate between the nuclear and cellular types. Free nuclear divisions occur, but cell wall formation accompanies nucleus division in some parts of the endosperm as well. The next one is endosperm hysteria. A remarkable feature of the developing endosperm is its capacity to take its nutrients from the surrounding tissue for its own development. Nutrient gathering outgrowth called hysteria may develop at either the micropylar and chaliceal ends and reach into the nucellar, integumentary or even ovary tissue. Hysteria often branch into several prominent lobes or divertically. When local food supply is exhausted, hysteria lobes terminate and become crushed by further endosperm and embryo growth. The mature endosperm. How a mature endosperm is present. Monocotyledonous endosperm usually reach their maximum morphological development and physiological maturity and remain to comprise a major part of the seed. In case of dicotyledonous species, the endosperm may develop or may be used up by the developing embryo and comprises none and only a small part of the matured seed. Seed with little or no endosperm is called as exalbuminous, and the seeds with having well developed endosperm or perisperm is called as albuminous. Some species have a well developed chalososperm in which both nucellus and endosperm disappear during development, and callosal tissue proliferates and forms storage tissue. The outermost layer of the endosperm is known as alluran layer, and it becomes thickened and filled with protein granules, and it functions as both a storage tissue and for secretion of hydrolytic enzymes, which upon activation during germination helps to break down storage tissue. Here in this diagram shows the comparison between this diagram shows the overall summary of the life cycle of the plant is depicted here, and this picture from the floral stem, from the flower, from the pollen sac, that is we have seen no archisporal cell, etc. Here the microspore mother cell is formed. Then from the pollen sac, microspores are formed, which is haploid in nature. From that, a mature pollen grain is formed by meiosis cell division. This is called Microgametogenesis, and here a pistil, from the pistil megaspore mother cell is formed, and which undergoes meiosis division and forming a complete embryo sac having ovary, egg cells, synerges and antipodal cells. Here from the sperm cells, two sperm nuclei are enter into the embryo sac via the microfile. Fertilization occurs. After fertilization, egg cell and one sperm nuclei combine to form a diploid fertilized egg is formed, that is zygote is formed. After that, the developing embryo and endosperm is formed. Complete after maturation of embryo and endosperm development, a complete dormant seed is formed as the cotyledons and axis occurs. From that, a root and shoot meristem giving rise to a seedling is formed. After that, a further development of organ formation, that is stem, leaves, roots, etc. are formed. Thank you friends, thank you for watching, and this video is highly useful for you. Maybe I think it will be very useful for you. Thank you for watching. Kindly subscribe our YouTube channel to get the upcoming videos. Kindly share, comment and like this video.